Okay, so hello everyone. So in the last lectures we looked at bacterial growth and disease and why it's really important for pharmacists to have a good understanding of these topics. So that was the end of our specific bacterial lectures, and today's lecture is going to be about the control of microbes for health and safety. So the control of the levels of microbes, especially pathogenic microbes, is crucial in a clinical setting like a hospital, especially areas such as operating theatres. And it is also important in other areas such as research laboratories to protect scientists and also to ensure they get usable results from their experiments. Just to mention that you have a microbiology revision session in a few weeks, so that's week 11, and this will be really important for your exams. So to get the best of out of these sessions, I really need you to email me with the topics that you are struggling with so that I can plan the session around these topics. So please email me over the next 2 weeks with the specific topics that you want me to cover. So by the end of this lecture, you will know why it's important to control microbes, especially in clinical and lab settings, and also the factors that affect microbial control. You will also know the different physical and chemical methods used to control microbes and why these are important. You will also learn the terminology around controlling microbes, for example, the difference between sterilisation and disinfection. And by the end of the lecture, you will have a good overview of the significance of controlling microbial growth and the various methods. So to start us off, we are going to look at the different terminology around microbial control. And this is important because it can be confusing. So the problem is that these terms, such as disinfection and sterilisation, are often used in inaccurate or imprecise ways, especially by non-scientists. So they will use a lot of these words interchangeably. But because you're going to be pharmacists, you need to know the specific definitions of these key terms and use them in the correct context. So first of all, we have sterilisation. And sterilisation is the killing or removal of all microorganisms from an object or habitat. A sterile object is totally free of viable microorganisms, spores, and infectious agents. A chemical agent used for sterilisation is called a sterilant. So that's a sterilant. Then disinfection, on the other hand, describes a process that eliminates many or all pathogenic, so that's disease-causing microorganisms, except bacterial spores on inanimate objects. So disinfection describes a process that eliminates many or all pathogenic microorganisms, except bacterial spores on inanimate objects. Disinfection therefore results in a substantial reduction of the total microbial population, but it does not completely sterilise an object because viable spores and a few microorganisms will remain. And then we'll talk about preservation. So with preservation, we are talking about the methods used to prevent microbes from spoiling food. So there are lots of different methods, such as canning, pasteurization, freezing, irradiation, and the addition of chemical preservatives. And these methods are used to minimize pathogenic bacteria, keep food quality at its best, and also to extend the shelf life of the foods. Then we have decontamination, and decontamination is a general term used to describe the destruction or removal of microbial contamination to render an item or environment safe. The term decontamination includes sterilisation, disinfection, and antiseptics. This leads us to antisepsis. So antiseptics can sometimes be called skin disinfectants. So these are the chemical agents that are applied to skin or living tissue to slow or stop the growth of microorganisms. Examples are antiseptic creams or liquids that you apply to cuts, such as Germaline or TCP liquid. Dettol antiseptic liquid is another example. Also the antibacterial hand washes, soaps and hand sanitizers that you see in Boots. These are also antiseptic preparations. And in addition, the alcohol rubs they use in hospital. These are antiseptic preparations too. And the last term on our slide is sanitization. So in sanitization, the microbial population is reduced to levels that are considered safe by public health standards. The inanimate object is usually cleaned as well as partially disinfected. So for example, sanitizers are used to clean eating utensils in restaurants. So this slide looks at where and why do we need to control microbial growth. So one example is it's important in the research lab to protect the scientists, but also to protect their experiments from contamination so that they get the results that are valid and usable. It's also needed in hospital for obvious reasons. So you will have patients with infectious disease and you do not want the infection to spread to other patients or to visitors and then taken outside to the community. You also have patients who are immunocompromised in hospitals, for example, chemotherapy patients, and if they cause an infection, it would have devastating effects. You also need to protect the healthcare professionals, such as doctors and nurses, for example, with masks and gloves, etc. And controlling microbial growth is important for the food industry. So as we said before, this is to destroy pathogens and to retain the food flavour, but also to extend the shelf life so the products last for longer. And then in the pharmaceutical industry, these measures are important to make sure the medical supplies they produce are not contaminated. And lastly, this is something we all know at the moment, we need to think about in our own homes, controlling microbial growth. So it's important that we clean and disinfect the house, that we wash our clothes, that we make sure we heat food properly. So this is in order to prevent us getting infections and prevent us spreading the infection to other members in the family. So a microbial population is not killed instantly when exposed to a lethal agent. Death is exponential, so the population is reduced by the same fraction at constant intervals. And then if you plot the logarithm of the population number remaining against the time exposed to the antimicrobial agent, a straight line plot will be produced, as seen below. The steepness of the killing curves depends on the sensitivity of the microorganisms to the agent, as well as on the agent's effectiveness. The survivor exposure curve drops at a steeper angle when heat is applied, and then at a flatter angle with radiation or chemical disinfectants. Another factor is the number of microorganisms contaminating a product, so we call that the bioburden. When applied to high organism concentrations, an antimicrobial agent will require a longer exposure time to achieve the same killing effect. So there are several different conditions that affect antimicrobial action. So first of all is population size. Because an equal fraction of a microbial population is killed during each interval, a larger population requires a longer time to die than a smaller one. The next factor is population composition. So the effectiveness of an agent varies greatly with the nature of the organisms being treated, because different microorganisms will have different susceptibilities or sensitivities to the antimicrobial agent. For example, bacterial endospores are much more resistant than vegetative forms, and young cells are more easily destroyed than mature cells. Also, certain species such as M. tuberculosis, which we mentioned in the last lecture, are more resistant to antimicrobial agents than a lot of other bacteria. So the next condition is the concentration of antimicrobial agent. So often, the more concentrated the antimicrobial agent is, the more rapidly microorganisms are destroyed. However, agent effectiveness is not usually directly related to concentration, so the relationship is not linear. And it is not always the case that the more concentrated the agent, the more rapid the killing is. For example, ethanol is more effective at a lower concentration of 70% than 95%. And this is because the activity of the ethanol is enhanced by water. So just to recap, although often the more concentrated the antimicrobial agent is, the more rapidly the microorganisms are destroyed, this relationship is not linear. And also this is not always the case. You have examples like ethanol, which kills more effectively at a lower concentration of 70% than 95%. And then the other condition is duration of exposure. So the longer a population is exposed to an antimicrobial agent, the more microbes that are killed, which is quite logical. Temperature is also a condition. A higher temperature will often increase the activity of the antimicrobial. So frequently, a lower concentration of disinfectant or sterilant can be used at a higher temperature. And then the last factor discussed is the local environment. So the environmental factors can either protect the microbes or enhance the action of the antimicrobial agent. For example, heat kills bacteria more readily at an acidic pH. Then you've got organic matter, which this is also an important environmental factor. Organic matter can protect microorganisms against physical and chemical agents, antimicrobial agents. So biofilms are a good example of this. If you remember, we talked about microbes forming biofilms in previous lectures. So biofilms contain organic matter, which will actually protect the microorganisms within that film. So we'll next just discuss briefly the mechanisms of action of the different methods of killing microbes. We'll go into more detail with each method later on. But just briefly, antimicrobial agents can be physical, such as heat and radiation, or chemical, such as ethanol or chlorine. And these agents have different methods by which they kill microbes. So when microorganisms are killed by heat, their proteins, their enzymes are irreversibly denatured. Whereas ionizing radiation results in the formation of reactive groups, and these react with DNA and affect proteins. Then exposure to UV light causes structural changes in DNA, and this prevents it from replicating. Then you have the chemical agents, such as alcohols, phenols, aldehydes, oxidants, and these denature proteins irreversibly. Then you've got surfactants, such as detergents, and these attack cytoplasmic membranes. And you've got acridine derivatives that bind to DNA and prevent its replication and function. So we're going to go into these methods in a bit more detail next. Okay, so before we move on to talk about physical and chemical methods of disinfection and sterilization, we're just going to mention about prions and archaea. So the standard sterilization methods damage bacteria, protozoans, which are often parasites, fungi, and helminths. Helminths are worms, and worm eggs. So these organisms, microorganisms, are damaged by standard sterilization methods. However, much more extreme processes are required to inactivate prions. So autoclaving at 121 degrees Celsius for 4.5 hours or at 134 degrees Celsius for 30 minutes. So they require higher temperatures and longer periods. And prions are infectious proteins. So these are the proteins that cause mad cow disease, which you might have heard of. And in addition, you've got hyperthermophilic archaea. So if you remember, hyperthermophilic organisms are ones that are heat loving, so live in very hot environments, and they can proliferate at temperatures of 100 degrees Celsius and higher, and they can tolerate autoclaving at 121 degrees for 1 hour. So these extreme life forms are not covered by the standard definitions of sterilization and sterility.